I'd like to start by reminding everyone that there's about 9 million species, actually probably 10 million species estimated to live on this planet. This is an underestimation. We have genomic information for about 2 million species, mainly from metabarcoding data. And all the mechanistic cell biology we know come from a 50 model organism, maybe 60 now. But, if we try and place all of these model organisms on a eukaryotic tree of life, they would mainly fall into plants, fungi, and animals. And if you struggle to find where humans are, they will be in bilitarian in this tiny line. So what does that mean? It means that we are missing out on a lot of amazing biology out there. We're missing out a lot on diversity of how specific processes are taking place across the tree of eukaryotes and the tree of life in general. And we're missing out a lot on evolutionary question because we just don't have all the intermediate, all the different species out there that will tell us about the historical or evolutionary process that happened, okay? So in my lab, we're interested about one of those big questions, which is the transition to multicellularity, how a unicellular organism decided to move and become a multicellular embryo-like structure. And this transition happened more than 50 times across the tree of life, and currently our focus is trying to understand how animal embryos first evolved. But we're planning to go explore this across different nodes of the tree of life. So I'll tell you first about this part of the talk, about what we do in the lab, and then I'll tell you in the end, hopefully if we have enough time, about how we're trying to reveal all the eukaryotic diversity with just more amazing imaging. All right, let's dive in. Sorry. So as you probably all know already, all animals look pretty different in shape and form, but they all share a common developmental process, which is you start with an egg, it undergoes fertilization, it starts undergoing cleavages to form a multicellular embryo, which organizes in space, and then you have some sort of a blastula, which undergoes gastrulation to form the future body plan. There's some differences here and there across the tree, but this is a general concept that is conserved across animals. Now, what we also know about all animals is that they evolved from a single ancestor. So the jump into multicellularity happened once, it's monophyletic, which gives us a very simple question, is how animal embryogenesis first evolved, right? Is it an animal innovation, or can we predict or find some sort of pathway that was there before even animals were there? So how to study this question? One of the ways to do it is to look at the closest living relatives. A bit like investigating apes to understand human evolution. Here we just leave the animal lineage and we look at the closest cousins. And you probably already have heard of first cousin to animals, which is chronoflagellates. These are unicellular organisms with a collar and a flagellum. And under specific conditions, where in contact of specific bacteria, they will start forming these multicellular colonies. You might also have heard about philosterians, which are the second cousin to animal. These are a bit different, but again, under a specific condition where they encounter a specific lipoprotein in the medium, they will start forming multicellular aggregates. But what about the third cousin? This is what I got interested in. But as many people here, no one cares about the third cousin usually. Why I ended up enjoying and starting working on ichthyosporins is just because they have amazing cell biology, and I'm going to show you a bit, a glimpse of what they can do. So in my lab, we took the bet of working on ichthyosporins with a very comparative approach. So we take the idea that animal embryogenesis is very simplistic. You have cleavages, spatial cell organization, and cell type differentiation. And then we go dig into all the ichthyosporins we can find in the wild or we can culture in the lab, and we try to assess whether we can find any of these modules and whether they can tell us anything about the evolution of animals. So for time's sake, I'll tell you two stories, two bits of a story today, one about Spheroforma artica, which is here on the tree, and then towards the end about Chromosphere perkinsii, which is here in the tree. And you can already see from their name, they're pretty different beasts. Spheroforma artica was isolated in the gut of an amphipod in the Arctic, loved the cold. Chromosphere perkinsii was isolated in sediments in Hawaii, so more around 27, 29 degrees. Let's start with Spheroforma artica. As I told you from the beginning, I'm a cell biologist, microscopist, so we start by imaging. So I'm just going to show you how they look like. So very, very round, spherical, they don't move. This is a 3 days movie. You know, they grow happily, nothing very fancy. And then at one point, you'll see they just pop out hundreds of cells. Of course, as a cell biologist, you know, you can understand how you cut a cake in two. How do you cut a cake in 128 pieces at the same time? Because it's 128 pieces every single time. So of course, one way to start figuring this out was to do simple experiments. So we synchronized the culture, and we did bulk RNA-seq. And one thing that we observed is that the first part of the life cycle, this is where we see all the mitotic machinery firing, whereas at the end of the life cycle, we see all the cytokinetic machinery firing. And that was already a first indication that led us over 5 years of work to show that actually ichthyosporins, or specifically Sphaerophora artica, undergoes what we call a synocytic life cycle, or similar to Drosophila syncytial embryonic development, where you start with a single cell with a single nucleus, which undergoes multiple rounds of nuclear division. This is timed every 7 hours. You duplicate the nuclei, so you form a multi-nucleated cell. And then at one point, you will reach a specific nuclear to cytoplasmic ratio, which will trigger what we call cellularization, which I'll show a bit in a second, to split every nucleus individually in order to repeat the cycle. So today, I'll just show you 2 bits of species. One is 2 bits of stories, sorry. One is about what mitosis look like, and then what does cytokinesis look like. Very simplistic, but you'll see it's very exciting. So if we talk about mitosis, so this is a project that was in collaboration with the lab of Gautam Deme and Yannick Schraub at the MBL, and led by Hiral Shah, who was a brilliant postdoc in both of our labs. So if we think about mitosis, the general view of animal mitosis, what we call an open mitotic strategy, where you lose the nuclear envelope integrity, you have centrioles making the spindle. Everyone knows that. This is what happens in all animal cells, right? But we have also the opposite view, where you have closed mitosis, which is most fungi undergo this, where you have nuclear integrity is maintained. You usually don't have centrioles. You have what we call a spindle pole body, which is a microtubule organizing center, which integrates within the inner nuclear membrane, and you form an internuclear spindle. So these 2 types of mitosis exist, have always been studied, and usually we say fungi do closed, animals do open, but we're talking about more than 1 billion years of evolution. So as these species are between fungi and animal, we wonder whether Sphaerophora artica is more of an animal in its division, or it's more of a fungi in its division. Okay. So first thing you do, you look at the genomes. We try to do comparative genomics to find if we can find centrioles, any markers of one or the other. And the answer is no centriole markers, no spindle pole body. We have no clue what the hell these guys are doing. Okay? So the only way to move forward is to image. We have no other way. So we had to develop all the pipeline to do ultrastructural imaging, be it by FIPSEM, where we already found this beautiful microtubule organizing center, which looks like a UFO, always sitting on the nucleus. And you can see all these magenta are actually microtubules emanating from the top side of the MTOC. This MTOC can transit from the outer nuclear membrane to the inner nuclear membrane when it's want to undergo mitosis. So you can see that it migrates by sitting on a nucleopore, goes through it, and then it starts forming the spindle inside. And the last method that we basically implemented and pushed in the lab, which is expansion microscopy, which you'll hear more towards the end. So this is a method which you embed your sample within a swallowable gel, and then you expand it physically. So you get better resolution, but in our case, it actually allows antibodies to go in, because in most of our species, most of the protists, microbial eukaryotes, have cell walls, theca, which makes it impossible to do immunostaining usually. But with expansion microscopy, actually open up and allows antibodies to go in. And when we did this, we can now observe microtubule. We can see the formation of spindle, which when you look at the ultrastructure of it and how it is forming over time, it's actually very similar to what we call an internuclear spindle, because we can see the spindle is actually all condensed towards the middle. And then we did TEM, and we can see that actually the nuclear envelope integrity is maintained throughout mitosis. So to summarize, Sphaerophora artica seems to undergo a fungal-like mitosis, closed mitosis, with using some sort of spindle pole body-like, but has none of the protein of spindle pole-like. So it evolved an independent mTOC that we're looking into right now. So what about cytokinesis now? I told you they basically divide every 7 hours. They will reach 128 nuclei, which is a specific number, and then they will undergo a trigger using the nucleotide-cytoplasmic ratio signal to allow cytokinesis. But how do you split them? And I haven't said that, but we don't have genetic tools, so you always have to be very creative in answering some of these questions. So we've been using a dye, which is FM464, which labels a membrane, and that's how it looks like when we image. So you can nicely see a progression of multiple membrane invagination around that starts from the outside towards the inside. They will progress at a specific speed. This is actomized independent, but also directed by microtubules. And what you can see is that it moves from the outside towards the inside, and then it will fill in the space, and that's how the cells then detach one from the other before splitting out. We've been also working on this process specifically, and we've shown that it's using actin and myosin to allow the building of this actin network. You can nicely see here in green actin. It's first looking as actin dots or nodes that will form actin filaments. This is only at the surface. You can see no actin in the middle of the cells. And then this actin filaments become actin cables. They know exactly where they need to go. They're surrounding the nuclei. And then they will start, with the help of myosin 2, to contract and pull in the membrane invaginations to allow the cleaving of each of these nuclei. What was exciting for us, and this is most of our work that we're ongoing work right now, is trying to understand this beautiful multicellular structure, which looks like an animal embryo. It is multicellular, it is polarized, and the question is how? Because again, there's many of the proteins that you maybe think of when you think about multicellularity that are absent here. So one of the questions is how this organism is able to do some sort of multicellular spatial organization. So to summarize very quickly, Spheroform artica looks and actually undergoes some sort of early embryonic development that's similar to the early embryonic development of Drosophila, using actomyosin, microtubules. It seems polarized, but there's also a lot of things that are missing. You know, mitosis looks different, as I showed you. I haven't told you, but there's no differentiation in the system, and then there's no cadherin, so have no clue how you can basically detach. Allow the cells to maintain together. So our vision of it is more of a convergent evolution of the same sort of process. Now, as I told you, we do comparative cell biology. So if we want to make general principles about ichthyosporins, we need to go look for other species to see if they do the same thing. So we started looking at chromospheric pregnancy with the same sort of mentality. We first do some movies. That was work mainly led by Marine in my lab. And you can see it's round, it grows in volume, there's some things happening, and then they will pop out. So at first glance, we said, oh, so it's the same. We can maybe do general principles, right? But very quickly, we started realizing there's some key differences. First, when we look at the nuclei, we see that these cells actually can go to up to 1,000 nuclei per cell. They're not synchronized. They don't divide every 7, 6, 5 hours. It's a bit random somehow. And then when we look at the diameter measurement, the diameter of the mother cell is actually always maintained, whereas the nuclear size is always going down, suggesting it's quite similar to what we see during early embryonic development in animals. So that was the first hint that there's something a bit different here. We again went back to a bulk RNA-seq, and that was with the help of John. You don't need to go through all the details. We're just going to focus on mitosis and cytokinesis. And here already a big surprise was that mitotic machinery and cytokinetic machinery are actually not uncoupled. They happen at the same time. So that was already a hint that we are basically exploring a different type of organisms. So we go back, mitosis, cytokinesis, okay? If we go back to this graph, same question, is it more like an animal or is it more like a fungi? First surprise, we found centrioles. First EM image was a 9-fold symmetry centriole. It was amazing. We do expansion microscopy. You can see that we have a beautiful metaphase plate. The spindles are like human cell spindles, totally different than the first bug, but more similar to all animals. And then when we do EM, we again see, we observe actually that the nuclear envelope integrity is lost. So now we have an organism that is third cousin to animal. that is undergoing animal-like open mitosis using centrioles. Pretty exciting. What about cytokinesis? Now, that was also the biggest surprise, I think, in my lab, because when we repeated the experiment with the FM464 dye, we actually saw this. And I'm going to run the movie multiple times and look at your faces. Always good. So what you can observe is that the cell is actually undergoing cell cleavages in an oriented manner. You go from a 1-cell to a 2-cell to a 4-cell, and actually it starts becoming chaotic after the 4-cell. One of the reasons for this is potentially the light toxicity that we're trying to work around, but it's possible that it's also a bit chaotic after the forced cleavage. That is, of course, one of the biggest similarities that you can observe with early animal embryos. But similarities didn't stop there. When we started looking using expansion microscopy about how the microtubules organize, how the actin organize, the nuclei, we started observing very exciting things. One is that before the first cleavage, what happens is that the nucleus, actually which is in the center, migrates to the cortex, and the first division happens at the cortex, very similar to what you can find in some ovocytes. The first division is asymmetrical. One of the 2 cells is bigger in size. One of the 2 cells actually undergoes mitosis earlier than the other. And for those who don't know, I'm pretty sure all of you know, but asymmetrical cell division is one of the main driver of cell differentiation in multicellular systems. Now, these cells keep dividing till they form, as I said, multicellular structure with up to 1,000 nuclei, and very often we see these empty cavities in the middle, which is very reminiscent of blastocell formation in animals. I'm not going to call this a blastula, because it's not an animal. So mitosis like an animal, cell cleavage is like an animal, there's symmetry breaking, very exciting thing. Is there cell differentiation? That was a very, I would say, lucky experiment where Channy was just testing all the antibodies in the fridge using expansion microscopy, and she came across these late colonies where you have, I think we have here 689 cell stage, and she was using this acylated tubulin antibody, which labels stable tubulin structures, and flagella is one of the stable tubulin structures. And what you can see for the volume is that, well, you can nicely see that within this multicellular structure we have flagellated cells on one side, and we have dividing cells on the other, and all the flagellated cells seems to be clustering on one side. And this is, we dug into this. This is quite repeatable. We can find always around 1/3 of the cells to be flagellated. They're always clustering in one side of the cell, hinting that we have spatial cell differentiation happening within the system. And when we look at the cells, we actually see always there's different cell types, which we suggest that at least more than 2 cell types coexisting together in the multicellular structure. We have flagellated cells, mitotic cells that divide in 2. We have amoeboid, or whatever you can call these, dancing cells, and then we have amoeboflagellate that are basically traveling around. So to summarize, we have a model which is third cousin to animal, undergoes mitosis like an animal, form multicellular structures, or at least early embryos, and then undergoes spatial cell differentiation. So we can argue at coffee whether this is the ancestor of all embryos or not. But for me, I think what is really exciting is that now we have 2 model systems. One undergoing development like a Drosophila, one is going development like a mouse, and now we can do comparative embryology outside of animals, and we can look at how division works out, morphogenesis, and differentiation, and then maybe one day we can figure out how animal embryos evolved. So now I'm going to tell you about the rest of the tree. As I said, we're focusing on this node now because everyone is interested in animals, but multicellularity evolved across. And we know, and we have thousands of cultures that are currently growing in culture collections. We have genomes for these, but most of you are not working on them. And one of the reasons for that is that there's no easy way to image these species. At least as a microscopist, this is how I argue my comment, right? And why is it hard to image these species? Because they have a protective cell layer, a theca, some have glass shells. It's really impossible to get antibodies go in, or even, genetic tools don't even think about it, okay? But we're not the first to start thinking about this question. There are people who have been trying to push some of these methods. You've seen maybe papers where people are trying 18S fish with antibody labeling, or using dyes to get overall structure from TREC or TAR data. But these are low resolution and they are low permeability because antibodies still don't go in. And on the opposite hand, we have, of course, more and more people doing FIBSEM to try and get full resolution. But these are extremely pricey, right? You get around 7, 8K for one cell volume. It takes months, maybe now weeks in some labs. And still after all of this, you don't have molecular labeling, right? You cannot use specific antibody, or it's not easy to get molecular identification on some of your proteins. So we were wondering whether expansion microscopy can help and can just expose all of us to more beauty and more eukaryotic diversity. So for those of you who don't know, I'm just going to make one slide about expansion microscopy. It's a method that has been developed now 10 years ago in the lab of Dudin. It's literally embedding your sample within a swellable gel. The swellable gel is made of, now there's many ways, but it's made of sodium acrylate at the beginning. Sodium acrylate is this molecule that you can find in Pampers. So if you add water on it, it will swell. And there are many, many protocols out there. If you start looking at expansion microscopy, I think we're beyond the 50 different protocols. But all of them, they will fall into two categories. One is pre-expansion labeling, where you first drop the antibodies, and then you cross-link the antibodies, and then you expand. This doesn't work for us. Again, there's a cell wall. Antibodies don't go in. So we've been mostly focusing on the post-expansion labeling protocols, specifically the ultrastructure expansion microscopy protocol from the Guichard and Amel lab in Geneva, where you first do the expansion, and there's specific steps for it, and then only at the end of the protocol, you add the antibody. Once everything is cracked and expanded, then antibodies go in, and it works pretty well. So as I showed you in my previous slide, it worked perfectly and probably saved my career when working on ichthyosporians. So I was wondering whether can we scale it up and do it on as many eukaryotic species out there? And that was a project that was held by multiple labs across two institutions, Paul Guichard, Virginie Amel from University of Geneva, Gautam De, Sinem Saka, and Yannick Schwab's groups at EMBL on one side, and then we of course partnered with amazing people at Culture Collections from Roscoff Culture Collection, it was Ian, and then Sergio Swan from the BMCC in Bilbao. And the idea was very simple. We went there and we told them, what culture can you give us now? And then we're gonna fix them, and then we expand them, and we're gonna just see if expansion microscopy works or not. They were very generous. We got around 231 species across what they had. Of course, there's still lines in the eukaryote tree that are missing, but across all of these, we managed to get almost 80% success for expansion across all of these lineages. And 70% or even 72% were staining with the antibodies that we had in-house, which is, it really gives us some sort of broad spectrum imaging modality if you're thinking about finding a new model system outside of human cells. Now, I don't have time to go through all of the images. I'm just going to give you a snippet of what we found. So one of the super cool things about microbial eukaryotes is that they do very finicky things that no one has had the chance to understand or look at. And one of the things that dinoflagellates do is that they do a very weird mitotic strategy where they make what we call a nuclear net, which is tubes within the nucleus. The microtubules go in there, and then they start popping out of these tubes to pick the chromosomes, and then they split the chromosome, which when you think about it as a cell biologist makes no sense. But this is what they do. And that was based on 2 studies, FIBSEM data on 7 cells, where they showed us that they do some sort of nuclear net structure like this. Because dinoflagellates don't stain well with antibodies because they have thicker, antibodies don't go in. No one has been ever able to get high resolution images of these. Well, now with expansion microscopy, we can do it. We can count them, and we can do it across tens of species. We can do it across hundreds of species that are positioned differently across the dinoflagellate clade, and we can potentially understand how this weird mitosis actually evolved in dinoflagellates. We have generated more than 1,500 volumes in less than 14 months across all of these species. Everything is open access. Go check it out. But one of the questions that I'm on time actually, I'm okay. I told you you're gonna have to stand up to pull me out from here, so that's fine. So there's 4 slides left, so I'm confident I'm gonna be fine. So what I wanted to say is that most protistologists actually go to the field, get samples from a pond, and then let it grow, and then you start imaging it, right? So we tried to do the same, and we wondered whether we can identify different species there. And that was from Tokyo Bay. You can see there's hundreds of different species there, and we wonder whether we can identify different species, and the answer is yes. We can find the ciliates. These are different types of ciliates. You can now actually get high-resolution images of these. You can find U-plots, which is also another ciliate, and Vorticella. We even pushed this, and you're going to hear more about this from Flora tomorrow, using or taking advantage of the ongoing, which actually it's not ongoing anymore. The analysis is ongoing, but we've been going for the past 2 years across Europe with the Tara schooner and this beautiful truck to try and get samples from the European coastline. And does expansion microscopy work? The answer is yes. You can see these are beautiful dinoflagellates from Barcelona. So now we're trying even to associate some of these eukaryotic diversity with, the European coastline. And just to finish, I want to tell you 2 things. One, this is all 3D volumes, and I want to show you. This is one of my favorite protists. I don't know if you've ever seen this. It's ciliate lacrimaria, single-celled, has a head and a beautiful mouth. It's actually scary. I think we should make a movie about it. It will open its mouth and it's just going to eat the whole thing in a second, right? And this is a species that grows in the wild, impossible to grow in the lab. No one has got a real good image, ultra-structured image of what does this mouth look like, right? Well, we got it. I'm just going to go through the volume. Microtubules will be in magenta, and membrane, more or less, ultrastructure of the cell is in yellow. You can see this is the mouth structure. And what is fascinating to me is the mouth. You can see, anyone has ever seen a microtubule vortex for a mouth? Anyone knows how you can build something like this? I think it's worth investigating. And to finish, so the preprint is there. You can check it out. And I just want to say that we recently got almost 2.5 million from the Moore Foundation for the next 3 years just to push this project. So we have money to teach you. We have money to test for you. We have money to image any microbial eukaryote you're interested in. And if anyone want to come to Geneva, we can teach you how to do this, okay? To finish, I want to thank, of course, the team. The team is growing. So we just moved to Geneva, so now we're 7. And I want to thank also our collaborators, the MBL and the Moore Foundation, Planning XM, and all of you for listening. I'm okay.